Welcome. I'm Doug Sesserman, privileged to serve as the CEO of American Associates, Ben-Gurion University of the Negev. I'm in my virtual office in New York. It's noon Eastern, and we have several guests from Ben-Gurion University. It's 7 p.m. in Israel. Thank you all for joining us. And for this live from Israel webinar series called Ben-Gurion Briefings. Today, we're having our second follow-up webinar on the breakthrough research that's happening at Ben-Gurion University. I want to begin by thanking Danny Shemovitz, the president of Ben-Gurion University, for his leadership, as well as Chaim Hames, the rector, and Jeff Kay, the VP of External Affairs and Resource Development, for their leadership of a leading effort in Israel that's multidisciplinary effort used with over 50 scientists and over 16 different projects. Some of them we'll highlight today, but we just appreciate your work. And I also want to thank my staff who's working remotely and so hard, led by Melody Mokhtarian, Ronnie Strongen, and my assistant Gina Ferdman, and Dana Ben-Benyamin, who does all the liaison work between us in New York and Israel. So today we have a great set of panelists, Professor Gabi Sarusi, Professor Nadav Davidovich, Golan Shohar, and Dr. Chris Arnoush are all our guests. We're going to start with you, Gabi, about your breakthrough research on a 2-minute potential testing device. So let me just tell everybody a little bit about you. You're the Deputy Director of Research at our School of Electrical and Computer Engineering. And your team has developed a nano-thin coating that will turn an invisible infrared light into visible light for creating night vision for glasses that allow people who can't see at night to see as if it's during the day. You've also spent time in the private sector and returned to academic life recently. And tell us about how somebody in electrical and computer engineering has helped discover a device that may solve for the immediate testing of COVID, which is one of the biggest problems we have. Gabi?
Yeah, okay, thank you so much. The basic idea starts from the fact that I'm completely not a biologist. I stopped studying biology when I was in 9th grade, actually because I had a female teacher and she left, and so I turned to physics. So I was looking at the problem of testing the viruses from the physics point of view, but not from the biology point of view. You know that there are two main ways to check or to test whether somebody is infected with the coronavirus. The first one that everybody is hearing today about is the PCR, which basically a biological test that you basically take some sample from the saliva, and then you, knowing that there are viruses there, you increase the amount of viruses and increase the amount of the RNA viruses, and then eventually, after a few hours, you have enough population of RNA in order to check them whether they are or not. So this process takes a long time, few hours, and there are some errors. Another way to check is to do a blood test, basically, in order to find whether there are antigen in the blood that shows basically that our body start to some immune to the viruses, and we are actually looking for the antibody. But this can take place only after 4, 5, or 6 days after the infection. That is that during that time, the doesn't know that he is infected, continue to spread the disease. And this is quite problematic. So the way that we are looking at the problem, I'm coming from the nanotechnology world. And basically I said to myself, let's look at the virus as a nanoparticle.
What is nanotechnology?
Nanotechnology is all the world that we are dealing with a nanoscopic structure of materials, namely materials that are in the size between 10 nanometer. 1 nanometer is 1 billionth of a meter. Okay, so it's a very, very small dimension. And the viruses, for example, their sizes, or the diameter of viruses span between 20 nanometer and up to about 400 nanometer. So they are quite tiny, unlike, for example, bacteria, which are in the size of micrometer, which is larger than viruses. And it is very, very hard to deal with viruses. And of course, it's better to see viruses because you need a microscope. So returning to
a virus is a much smaller, is much smaller than a bacteria in terms of its size.
Yeah, much smaller, yes. Much smaller than a bacteria. And therefore, but lucky us, is that we are dealing all the time in the Nanotechnology Institute at Ben-Gurion University. We are doing all the time with this kind of sizes. All the work that we are doing are related to nanoparticles. So we said, let's treat the viruses as we treat the nanoparticle that we are working with on daily basis. And the idea that we came with is actually saying the following. Let's measure the existence of the viruses on a certain surface. And the way that we are able to do it is that we are putting the viruses on a very small, what we call it, micro or nano antenna that we are putting on a surface of a glass. And basically what happened is that you know that any antenna, you have a frequency. If you are looking at TV, you remember that you had to adjust exactly the channel. Each channel has its own frequency. The same thing is with antenna, very small antenna, or actually nano antenna. It has its own specific frequency, and this frequency actually can tune by the size of the antenna or by the material that the antenna is made of. So the way that we treat this issue is that we try to see whether we can make a very, very small and tiny antenna that we will be able to resonance in certain frequencies. But if we will put the viruses on top of this, it will change the frequency, the resonance frequency of the antenna. Namely, it's like moving from channel 5 to channel 6 on the TV. And that's exactly what we are looking for, whether we have a shift of the frequency of this nano antenna, very, very small antenna, from channel 5 to channel 6. And, and once we will see that there is such shift of the frequency, we know that we are detecting actually the coronaviruses. So these frequencies, since we are dealing here with a very, very tiny feature or creatures, the viruses are very, very tiny. They're not really creatures, but very, very tiny samples. We have to build a very, very tiny antenna that basically will match their sizes.
And Gabi, my understanding is that this antenna is a sensor that could be on a handheld device where people could actually be tested in large numbers, say they were going to a sporting event or when life starts to return back to normal, and within a short period of time, we would know if they were positive. Is that correct?
Yeah, and that's exactly what I want to refer to. The nice thing about, first of all, we found that it is possible. What I said is possible, and we proved in my lab that we can see those shifts using the viruses. And we saw the resonance of the antenna, let's say on channel 5, and when we put the viruses, we saw that it shifted to channel 6. And that's exactly what our simulation said. And basically we confirmed that what we had in the simulation actually we can see in the lab. And we were very, very happy to see it. Now, how to implement it, and this is refers to your question. If we want to eliminate this pandemic, we have basically to check the entire population in the world. Why? If we will do it, we'll be able to put in quarantine those who are infected, and all the other can go to work and so on. But we cannot do it because we are not able to make a rapid test the entire population. Why we cannot do it? Because it takes a long time, and it's also very, very costly. So our method, we believe that if it will be automated, it can take something like between 5 and 10 seconds. Right now, we don't say it. We say that it will take 1 to 2 minutes. But I believe that if it will be completely automated, it can take seconds. And
Wow.
the basic idea is to make those tests not at the point of care as they are done today, but at the point of entry. Namely, if you want to get to the factory that you are working, to the company that you are working, to the building that you are working in, you would make this rapid test, and then it will be confirmed to get into the building, or to be authorized to get into the building or not. And this is a major, major difference from what we have today. So this is our vision. By the way, it's related to Corona right now, but it can be done for any kind of viruses, because we found out that each virus has its own signature, namely one virus will shift the frequency from channel 5 to channel 6, and other virus will shift the frequency from channel 5 to channel 7, okay? And this is happening with different viruses because different viruses have different sizes. So we can differentiate with this method by different viruses. And this is the nice thing about the method.
So if I understand this correctly, your research could lead to not only enabling us to know whether we're sick and carrying it or not, but also to help us get back to work and society where people can go to events, et cetera, and not be worried about this contagious virus. So one last question, Gabi, how long till this is commercializable and might be on the market?
Yeah, right now we are a very, very small team, and I'm using a lot of volunteers because most of the university is shut down. So I'm getting authorization to ask the people from the nanofabrication to do the chip for me, because it is based on these tiny chips. And so I'm getting a lot of help from friends, a lot of friends from people that I know, and we are pushing it ahead. The nice thing is that within 2.5 weeks since we started about pushing this idea, we had full simulation of the device. We had a lot of devices that up to now we produce something like 500 chips in the fab, and we already order a system to come to Israel from Germany, and it arrived yesterday. And yesterday night, the system was fully operation. And today morning, we already started checking the viruses, the viruses on the chip, and making several tests. And we already got Helsinki, namely authorization to do a test on human being in Shiva Hospital in Israel right now. So all this process we did within 2.5 weeks.
Thank you very much, Gabi. For the participants, and we have about 200 of you on this webinar right now, thank you for joining us. We're going to do a bit of a round robin conversation and then open it up to questions. I can see on the Q&A function that questions are already coming from the audience. If you are participating in this webinar and have a question for any of the individual panelists, just click on that Q&A button, write your question, and later in the program, I will look at the questions and then direct them to the appropriate panelists. I want to pivot now the conversation to Dr. Chris Arnouche from the Zuckerberg Institute of Water Research. You've been with us since 2012 at Ben-Gurion University. And for the last 5 years, my understanding is that you've been working on a self-sterilizing mask that uses a graphene membrane that can be used for other antiviral reasons, and it has direct applicability to the coronavirus. So Chris, why don't you explain your research?
Okay, thank you. I'll just start by just describing the material and how it came about that I started working on this material. But so basically, I made a collaboration with someone in the USA, and he introduced me to a new technique to generate graphene on many types of carbon surfaces. So this new technique is called laser-induced graphene, and you basically, all you need is a laser. It's a very low-tech laser. It's a laser that is in any laser cutter. So a very common laser. And you use that laser to treat surfaces, treat polymers, and it converts the top part of the polymer into like a 3D carbon graphene foam. So I chose for my wallpaper behind me here, I chose a picture of what this laser-induced graphene is. So down here in the lower part of the screen, it's a water treatment membrane. So it's a polymer. And on the top part of the screen, it's after laser treatment. So you can see that the laser converts the polymer into a really porous 3D foam. And this carbon material, this porous carbon foam, is electrically conductive, and it's permeable to water, it's permeable to air. And so for the past, let's say 5 years, I've been working on these laser-induced graphene surfaces and understanding what they could do as a filter. So I'll just share my screen now because it might be more, let's see, illustrative. 1 second. Sorry for it. Let me just share my screen. So hopefully everyone can see my screen. And so we think this laser-induced graphene could revolutionize.
You can see it perfectly. Thank you, Chris.
Okay, great. Both air and water filtration. So what is this laser-induced graphene? Like I said, it's you take a laser and you treat any polymer surface or most types of polymer surfaces. So here I've got polyimide. And what we discovered at first is that by itself, this material is very anti-fouling. So it means that bacteria, they don't like to grow on the surface. And so this lower picture here shows the bacteria coming towards the surface. It only grows on the polymer, but not on the laser-induced graphene. But because this is an electrically conductive surface, we connected wires across it. And this is in water, so they act as electrodes. And so with a charge and a small current going through this system, you can actually then get also active killing. So you get both of a passive anti-biofouling and also an active killing of bacteria. And we extended it to many different types of polymers. So here you can see polysulfone, polyether sulfone, and these polymers are very, very important in especially water filtration. Many, many water filters are made of this material. So when we're able to generate this 3D porous surface on water filtration membranes, we're able to take a water filtration membrane and have it in this like stacked conformation, so that when you're filtering water, you're killing the bacteria which are passing through these membranes. And so recently, we've also tested for viruses, not the coronavirus, but another vaccinia virus, that's a smaller particle. And so also, when viruses pass this filtration system, you also get a deactivation of the viruses. So we're very, very excited about this this filtration technology that can both kill bacteria and inactivate viruses. And so this technology has been patented, and a startup company is starting to look into what are the first products that could come out of this material. And so, as you can see here, I'm just playing a short movie. This is the laser which is passing over the filter material. And as it passes, you can see that you're starting to get this black coating. This black coating, I'll just skip it ahead a little bit. This black coating is this laser-induced graphene coating. And so one of the main steps that this company is doing right now is scaling up this process so we can generate this material on a large scale. So this is just in my lab. You can see it. You can make a small surface in a couple of minutes. And so the main idea is to get it on a larger scale. So the first products we're looking for, my collaborator showed the same material in an air filter configuration. So we've got this material both as now a water filter that can kill bacteria and viruses in a water environment. And also it was shown to be killing bacteria in an air filter configuration. So our first products, we're really looking for this active air filter, which can actively deactivate bacteria and virus particles to combine with, for example, heating, ventilation systems, air conditioning systems. So to create safe spaces in buildings, on airplanes, in cars, to be actively using this electricity and this graphene on
So the
filters.
application of your research, Chris, is far broader than just on a mask to keep a virus from getting in or getting out.
That's right. You know, masks, there's many types of masks out there already. So this material, you know, might be able to come into a mask, but it's more broad. We're looking at the bigger picture, getting safe spaces in airplanes, in cars, in elevators, in buildings. So that is where the company is headed right now.
Okay, thank you. I'm gonna share a different screen. So this, I believe, is the mask with your graphene membrane that people can see. And just tell us a little bit about how it is different than what we hear about the N95, or obviously a cloth mask that we're being told to wear in New York and other places around the US has none of these properties. So just help us understand how this is different.
Right. So this the company they're in a very early stage right now. The company that is starting to license this technology, and this was one of their concepts. Their concept of a potential product. So the you know, a mask, its main, I guess, action is one is if you get a good fit on the face so that you don't get leakage around, but as well to make sure that the person handles the mask properly. So if you, for example, get the mask wet or you get the mask is contaminated and then you touch the mask, well then the contamination is then on your fingers, which can go to your mouth, your eyes, and on surfaces. So because this laser-induced graphene is an active component, we hope that we can put this active component in different applications so that bacteria and viruses are inactivated as they touch the filter.
All right, thank you very much. So we're going to switch now the conversation to Professor Golan Shohar and Professor Nadav Davidovich. Golan, you're a clinical psychologist, and Nadav, you are the director of our School for Public Health, and the two of you have been collaborating on the impact of the coronavirus and also researching policy decisions by the government on the psyche of society, or how anxiety builds or handles the human condition can handle a pandemic or this kind of crisis like we've never seen before. So maybe Golan, we start with you. Nadav, jump in wherever and tell us what you're learning.
Okay, thank you. I'm happy to be here with you. It's true that Nadav and I are collaborating. I'm going to be briefer than the other speaker because we're essentially sharing the same information. I'm sure the viewers are aware of the fact that in this ongoing global crisis, public's behavior is paramount. It's paramount in terms of mitigating, containing, and coping with the virus. Prior to this, to our going live, Doug alluded to the fact that New York inhabitants are not very much accustomed to taking directions and abiding by instructions. So it's a good illustration of that. Both Nadav and I have been studying public's responses to mass medical crises independently, but this COVID-19 crisis united us. And so we have launched what might be one of our most important studies, examining the Israeli population even before COVID-19 infiltrated Israel, and then weekly, 7 weeks in a row, and we're still collecting data. I think that there are two behavioral aspects that are paramount in terms of public's response to the virus. One is anxiety, and the other is public's response and attitude toward the government or the agencies in charge of managing the crisis. I'm gonna leave the second one to Nadav because this is his expertise. Being a clinical and health psychology, I deal with anxiety a lot, so I'm gonna talk about anxiety. I'm gonna be brief, but before I do that, allow me to share a screen. Is this seen? Can you see that?
Yes, we can see it perfectly.
Okay. So in terms of anxiety plays a major role in mass medical crisis in general and plague or pandemic management in particular. Anxiety is an important outcome of any mass trauma, and definitely a pandemic such as COVID-19 can be considered a mass trauma. People tend to react with anxiety, and we already have predictions that there's going to be a mental health pandemic shortly after we get a hold of the virus, a mental health pandemic stemming from the economic crisis and from other issues.
Go on. Do I read this chart correctly, meaning that at week 0, there's a score of 2. So everybody's always a little bit anxious. Is that right?
Absolutely.
And then through this pandemic, the anxiety has increased by 50%. In the States, it just seems so much greater.
Well, exactly. You're reading the chart correctly, because what we are finding is that anxiety does not soar in the Israeli public. And I'm going to elaborate about that shortly. I just want to say two things. One is that one target population for anxiety, a target population that is incredibly vulnerable, is medical teams. There's now a new paper in JAMA suggesting that medical teams suffer from a host of mental health conditions related to their treating corona patients. Another thing that I want to point out is that too little anxiety is also not good. That complacency, as we have arguably seen in some of the countries in Europe, can be devastating as well. And so seeing a flat line reflecting very low anxiety that is steady is not good news. Anxiety is an evolutionary response, a proper evolutionary response to threat. And we would expect it to rise as the threat rises. The question is how much? And now let's look at the chart. And if we look at the chart, we will see up until week 4, a linear growth in anxiety, but one that is modest. How do I say that it's modest? First, if you look from one assessment point to the other, you will see an increase that is not formidable. And second, the line is not terribly steep. But even more importantly, the scale runs from 1 to 5, where 4 and 5 are really, I have anxiety. And never in this assessment period does the average level of anxiety touch the alarming values of anxiety. Moreover, you see that from week 4 onward, there is a plateau, there is a flattening of the curve, so to speak, that we would very much want to see for the virus spread. We see it in anxiety, and even some very, very preliminary indications that anxiety decreases. Now, why is that important? It's important for 3 reasons. First, prior to the entry of COVID to Israel, all the media was talking about an impending panic. And myself and other experts, Nadav as well, argued that there's no panic and we don't see any indications for panic, but the media didn't want to embrace that. This is a research evidence suggesting that there's no panic. Panic doesn't look that way. 2, it's good that the anxiety is soaring, because now we have in various quarters of Israel some cities in which complacency roamed, and the consequences are devastating.
This is what we hear about in Bnei Brak and other areas.
Exactly. The consequences are such that we're now having a full-fledged lockdown, because people weren't anxious enough and weren't staying in their home and were spreading the virus violently.
And for our participants who aren't familiar with Bnei Brak, that would be one of the neighborhoods where you have a large proportion of the ultra-Orthodox that live there.
Right, and the third thing is that you see a flattening of the line. So not only is the public not panic, the public appears to be adjusting. There is a steady state. Now, there is one more finding that I haven't shared with Nadav as yet, because I ran these analyses last night, very early at night, and that is religiosity. Actually, I did email that to the group, I think, Nadav. Religiosity predicts the flattening of the line. So our study, in a way, predicted what happens in Bnei Brak now. I'm going to stop here and leave the rest.
The way I would interpret that is, if you have a deep belief in God, you can handle God-created crises better than if you don't, perhaps. But let's switch to Nadav. Let's switch to Nadav, because we're beginning to run out of time. Thank you very much, Golan. My Q&A button is blowing up with questions, so we've got to give the audience a chance. Nadav.
Hi, first of all, thank you for having us. It's really a great pleasure. And I want to share my feeling of solidarity with the people in the US and especially in New York. You're going through a really hard time. We need to remember that Corona pandemic is very unique because we don't have still vaccine, we don't have medications yet that were approved by randomized clinical trials. So what we call the non-pharmaceutical interventions mainly the physical part, quarantine, isolation, all of that are very, very important. And this is why we need to see also the question of adherence and the psychological aspect. I really want to thank also Professor Golan Shachar, because he was so great in grasping the opportunity and being so quick to start. And the fact that we have the time zero is probably, we are one of the only countries in the world that we can get now this kind of development, the different measurements and the dynamic, because I think that what is also unique here, that we have a very dynamic situation with changing instructions, changing in the atmosphere, because just try to think, Doug, the situation in New York about a month ago and what's happening now, and just think that we have now 2 months of observation. This is unprecedented, and we still need to digest, I think, Golan, the treasure we have here. I just want to say that I'm also very interested in the question of trust, because trust is another factor influencing people's response. And we have the initial trust in the government that, by the way, was quite high. If you remember, Israel was one of the first countries in the world that blocked the entrance from other countries. And probably right now, this is the main factor why in Israel we are facing a relatively low R0 and relatively low numbers of people that are infected, etc.
Yeah, I think you just know, how your government knows and your population knows how to handle war of different kinds and crises better than we do. And certainly from an anxiety level, you've gotten used to a life at a certain steady state of anxiety, which we may have never experienced. And so that curve that Golan was showing was so much flatter than the steep thing we may be experiencing. It's incredible how life in Israel is different than life in the States.
You're absolutely right. We are now entering also in another phase of doing comparison to other countries, because the World Health Organization also started the study that we joined. So it will be really interesting to see this comparison. But I want to bring in also some other variables, because right now we were talking more about the individual anxiety level. And another thing we know, and Golan did research on that, and also co-investigator Professor Limor Aronson, the vice president of BGU. She's also in the School of Public Health and director of Prepared, the center dealing with preparedness. And this is community resilience. So that's another factor. And we saw that community resilience is also very important in terms of adherence. Other very important factors, of course, age. And we saw actually, and this is very interesting, that maybe in contrast to what people will think, that actually the people that are older are less anxious than those who are younger, even though we know that in terms of mortality, morbidity, age is a major factor, but probably life experience and maybe also the economic factor, is also very interesting here. Religiosity is another one, and later on,
although there's a very famous YouTube video of a 3-year-old Israeli that is handling the situation quite well. I don't know if you've seen that, but she's
I remember from the beginning. So just to summarize, this is a very unique study. We are now just working like crazy. Hopefully, we're sending it to Lancet. Golan, can I say already? And this will be, it's a gold mine. We have so much data, believe me, that we can continue. And it's important, both of the short term, trying to help now the government dealing with the situation, but also later on, because unfortunately in Israel, but all over the world, because of many other factors, including climate change, etc., this is not probably the last time we're going to face this kind of challenge. So I think that this combination of having someone like Golan from psychology, myself as a public health physician, Limor as an expert dealing with emergencies,
Yes.
Professor Itamar Grotto, by the way, who's also taking part, is the Deputy Director General of the Ministry of Health and also a faculty member at the School of Public Health, and other people. So we have a very unique interdisciplinary group, and we can take a look on this very interesting, fascinating topic of non-pharmaceutical interventions and behavioral aspects. By the way, the NIH, the National Institute of Health, took the question of behavioral sciences as one of the first priorities because we don't have yet vaccines, because we don't have medicines. So we, of course, will contribute to the ongoing discussion, and it's both important from theoretical, but also from very practical perspective. So I really want to thank Doug having us here presenting, and also the president, because he is also now supporting this study. And as you mentioned, Doug, at the beginning, it's part of the task force from Ben-Gurion University. I think we were the first university in Israel to start this kind of initiative on the university level, not just as individuals, and just connecting us all in different groups, studying different aspects, and showing the strength of Ben-Gurion University and Israel, of course, in large. So thank you again.
Thank you very much. So we have about 5 minutes left, and I want the Q&A is blowing up, as I mentioned before, so I wanted to go to a couple of those questions, and they're directed a lot to Chris and Gabi. So I'm going to ask you, Gabi, a question real quick. This is coming from David Breslauer and some others. What's the testing sample that's used for your device? Is it breath, skin, saliva? And if governments around the world could get behind your technology, how quickly could it be rolled out?
Okay, so the test that we are using is actually a saliva, and it's done in two different ways. One is to take a swab of the saliva, and the other one that we are developing right now is looks like a breathalyzer, like you know, when testing the alcohol level in the blood. So it's more or less the same, and that will be a really point of change in how to treat the testing of the coronavirus.
Very good, thank you. And quickly on if governments got behind your technology in a huge way, how quickly could it be scaled up and rolled out?
Yeah, okay. I will not be able to say everything right now, but it is scalable, but it is scalable in terms of month to up to 1 year, but not for the entire population of the world, but definitely for the population of US, population of Israel.
Okay, thank you. Chris, we're getting a question about the graphene versus like a silver nitrate or other properties that could be induced into your material, just in terms of efficacy and fighting bacteria or viruses. Could you comment on that?
Sure, yeah. This the material, the laser-induced graphene material, is very versatile, meaning that you can there's all sorts of different mechanisms on how you would be able to kill or inactivate bacteria and viruses. And like you mentioned, for example, you can put different metals also into the graphene. So like you mentioned, silver, you can put silver nitrate onto the surface, and when you put the laser onto the surface, you convert the silver nitrate into silver metal nanoparticles. So you can both get this porous graphene electrically conductive surface with embedded silver nanoparticles, which which is known to also be really, really antimicrobial and good to inactivate microbes. So it's very versatile, and there's a lot we can do with it.
All right, thank you, Chris, and thank you, Seth Perlman, for that question. There are a number of other questions that have come up. Unfortunately, we're not going to be able to get to all of them. Sissy, I saw that Sissy Zoller asked a question about Golan and Nadav's research was, have you done any research with the US population? And your research is really with the Israeli population, correct?
We are, Nadav is very strongly involved with the World Health Organization and the European countries, so he's going to look to that. I have stronger ties, I have more ties with North America, so we're collecting data in US and Canada as well.
Yeah, people here were trying to understand what was the panic buying for toilet paper. Was that because people thought we were going to run out, or was it to control something in an environment where you've lost control, so you can control what you hoard?
Do you have a
what's
panic?
the psychology behind that?
Yeah, it is an essential good, and it does signal anxiety when there is an overbuying or riots outside supermarkets. We don't have that in Israel. Do you have that in the States?
Not quite, but I'd say on the streets it feels quite surreal, and there is a very high level of anxiety, much higher than the charts I think you're showing.
We moved in Israel
your population. Go ahead, Nadav. I want you, Nadav, take the last word, and then I'm going to close it up.
Sure. I'm just saying that we moved from toilet papers to eggs, but this is because of the Passover. So just end by saying that you saw here the strength of Ben-Gurion by moving from basic science to biology, to public health, psychology. So it's really great, and thank you for the opportunity. And just to say that Corona will be over, but what we're doing now will stay with us. And the solidarity in Israel right now, in terms of, at least in the Negev, we're doing lots of work with the Bedouin community, with the Haredi community. So just to reflect also this part, Doug, is very important for me. We're doing lots of work with the communities in the Negev, and that's part of our mission as well.
Thank you. So I just want to wrap up with two thoughts. One is David Ben-Gurion was famously quoted as recognizing that Israel at the end of the day would be judged by the world, not by its military might or its technological achievements, but by its moral contributions as a light unto the nations. And I think this webinar and your great work is a perfect example of how technology and innovation enables us to be a light unto the nations. So thank you for all your work and for the amazing responsiveness and proactivity of Danny Shamovitz and the team at BGU to pivot all this great scientific work in the area of combating coronavirus. I see, Nadav, that you have the glass raised. We're on the eve of...
We are going to have a celebration before Passover on Zoom with the School of Public Health, so prepare the glass of wine.
Beautiful. So as people know, one of the most celebrated holidays of the Jewish calendar starts tomorrow, the Passover holiday tomorrow night. And as you may remember, it took 10 plagues in Egypt to free the Jews and make us no longer slaves and enabled to do the things that we do today. And we thank Hashem for that. Hopefully today in the year 2020, it will take just this one plague of COVID-19 to heal the world, enable us to see beyond our differences, to share this great innovation and contributions to Israel, which so many people constantly are complaining about, to showcase the amazing work that you do, the contributions to society in your own country, here in this country, but importantly in the world. So thank you all. We wish everyone a Chag Pesach Sameach, a happy Passover. And no matter how people are celebrating this year with virtual seders, connecting families and communities all over the world, we wish you the very best. Be well, and stay safe. Thank you very much.
Thank you.
Bye. Pesach Sameach. Thank you.
Happy
Bye-bye.
Passover.